Good afternoon, everyone. Let your microbes in your guts get to work digesting. My name is Ben Taylor. I'm an assistant professor in organismic and evolutionary biology here at Harvard University. And it is my great pleasure this afternoon to introduce to you Dr. Nicole Gerardo, who is a professor and director of the Graduate Division of Biological and Biomedical Sciences at Emory University. Dr. Gerardo's lab focuses on the evolutionary ecology of interactions between microbes and their hosts. She and her lab are interested in how both beneficial and harmful microbes establish and maintain relationships with their hosts. Such associations are, of course, shaped by ecological limitations of both host and symbiont ranges, evolutionary trade-offs for both the hosts and the microbes, and host immunology. And much of Dr. Gerardo's work utilizes the versatility of systems where both the hosts and their microbial partners and pathogens can be maintained in a laboratory setting. And through both genomic and experimental approaches, Dr. Gerardo and her lab address a real wide variety of topics ranging from the genomic diversification of the parasite in a fungus-growing ant symbiosis to the effect of field-sprayed antibiotics on bee foraging, behavior, and pollination in orchards. So it's really, really interesting work that's going on in the lab. Outside the lab, Dr. Gerardo has purportedly canoed, that's right, paddled a canoe from one side of a country to another. TBD on which country that is, you'll have to ask her. I'm guessing it's not this one. And she also teaches her research to monks. So fascinating stuff both in and outside the walls of her lab. So today, Dr. Gerardo will be telling us about the many steps taken in forming an animal-bacterial symbiosis. So without any further ado, welcome Dr. Gerardo.
All right, thank you so much. I know your gut microbes are telling you that it's nap time now, but it's not. My goal is to take you on a journey of a painting. So this is a painting by Peter Nicholas, who's an Emory undergraduate. And I want to tell you the science behind this painting and also tell you a little bit about its creation. We're going to start in the ocean. This is a squid. Some of you know this beautiful little creature. They're super cute, bobtailed squid. So this squid needed to go and just take in tons of seawater to find its bacterial symbiont. Those bacterial symbionts get in, and then through a lot of selection processes, end up establishing at really high density in an organ within the squid. And at high density, they glow, which actually makes it harder for predators to see the squid in the moonlight. This is a fungus-growing ant. So ants millions of years ago became farmers of fungus, and they decided, let's not leave it up to chance and just try to go find it every once in a while. And so when a queen goes to start a new colony, she actually stuffs a little pellet of fungus in her mouth. Maybe some of you did this with lunch today. And then she'll spit it out, and she feeds it, and then as there's more workers, they take care of it. So generation after generation, they make sure that they have their fungus by taking it with them. This is a pea aphid. Pea aphids were a sort of long-standing model in my lab and have actually been just sort of the canonical system for studying insect symbioses. As she is giving birth to her daughter, they're parthenogenic. They're all female, so it's a daughter. Before it was ever born, when it was actually very few aphid cells, that sort of little envelope of aphid cells was flooded with one bacteria called Buchnera aphidicola. The aphids have to have it. It produces amino acids they don't get in their plant sap diet. And so because it's so critical, we assume that that has just shaped the evolution of this organism to maintain this amazing process to pass it on to those embryos. And in fact, as this aphid is born, it has embryos inside of it that are already developing and are already full of Buchnera. This transmission processes are just really critical to shaping both the ecology and evolution of these systems. And we often think about how they're shaping the evolution of the hosts, which I'll tell you a little bit about today, but they're certainly also shaping the evolution of those microbes as well. So this is our host of interest. They're squash bugs. They are not as cute as squid. I'm willing to admit it. They are major agricultural pests here in the US. And in fact, in Georgia and many parts of the US in the summer, they eventually pull their squash plants in organic fields because there are just so many of these bugs that they just collapse the plant. So they feed on plant sap, xylem and phloem, and they hatch out in high numbers. And then, I don't know, until recently, I didn't think they did anything interesting, but I'm going to tell you something interesting. What we're going to talk about is sort of how they pick up their symbionts and what the consequences of that are for both the host and those bacteria. This story is absolutely sort of founded by the people in my lab. It is an amazing team, and I love the people I work with, and I love the fact that they support each other so much. In particular, I'll talk today about a lot of data collected by Jason Chen and Anthony Juncker, who are up here in the top right. Yeah, that's the right for you. But their work has really been sort of supported by lots of hands-on time in the lab by others, and also benefited from foundations set by others in the lab. Okay, this is a bug. This is a public service announcement. You are microbiologists. You say you work on bugs. A bug has 6 legs, and it does not have a flagellum. Do we got it? Okay. So this is a squash bug adult. What we saw before were the first instars. They had just sort of hatched out. If we take this adult and dissect out the gut, there's a foregut, and then there's a midgut of multiple sections here that are labeled sort of in 1, 2, and 3, which are all smashed together. I'm a really good microscopy person. And M4, which is this really sort of classical, sort of highly invaginated organ. The foundation of our field, the Darwin of animal symbiosis, was Paul Buchner. And he drew images like this in many bugs and in many insects. We see some kind of organ like this, some kind of thing along the gut. And occasionally he could sort of break it open, and you could see that there were a lot of bacteria in there that looked like the same thing. And so he postulated that these were organs that had been evolved to store bacteria at high numbers. They're associated with the gut. They're probably nutritional. He was not an experimental biologist, but he was an amazing natural historian and observer of the natural world around him. And like Darwin, he was right most of the time. So we can use fancy sequencing technologies and do sort of 16S profile in the communities of these different parts of the gut. If we look at the M4, it is dominated by these blue taxa, which are Caballeronia. These are bacteria that were until recently were Burkholderia and have been reclassified as Caballeronia. We see Caballeronia in other parts of the gut, but they're at very high density in these crypts. And what is amazing for me is when I was working on aphids, and I had this opportunity to work with great people and to learn a lot of tools, but Buchnera and most of the aphid symbionts that we work on cannot be cultured easily outside of the aphid host. I'm an evolutionary ecologist who likes species interactions, and I could really only see one of my species. And the great thing about this system is that Caballeronia is cultivable, which means we can do cool things, like we can fluorescently label it, and then we can more easily track transmission and see where it is and what it looks like, and this is what it looks like. This is Caballeronia in the crypts of the bugs. This is not photoshopped in any way. So Jason fed a bug of GFP-labeled bacteria, and then all he did is peeled off the cuticle on the top and then took this picture under fluorescent scope. So they're in very high density in these organs. If we zoom into that organ, what we see is it's a lumen and a series of crypts. So each of those crypts is a little sort of population of bacteria. We have no idea, really, about the details about what's happening in each of those little communities. Can the bacteria in one community sort of go in between these different crypts? How much metabolic differences are there between the crypts? I don't know. I want to find out. This is what I love about the system. There's so much to figure out. Because we can actually work with them so easily in the lab, we can do some basic experiments, right? We take some bugs, we feed them on diet with RNA or without, and then we can track things like host phenotypes and see what happens. And the first thing that we see is that survival is much higher when you have RNA than when you don't. Through a lot of experiments, it does need to be RNA. So we can go to other Burkholderia and see no benefit. In some cases, those can't actually get into the crypts, so that's probably it. The symbiosis doesn't establish. And in other cases, they get in there, but we don't see this sort of stark survival benefit. It doesn't matter too much what strain of Cavalironia it is, but we have found some strains that seem to provide less benefits than others, which is a whole other sort of arm of our research. Now, if you're an insect, your goal is to just get to be an adult and mate and hopefully lay eggs if you're a female before you get eaten by a spider or stepped on by a farmer. Like, your life is very simple, but you really need to sort of get through this developmental life cycle. So for those of you who are microbiologists, these bugs, we're gonna hatch out the eggs and they're gonna have to go through a number of instars, which you'll see sort of across the rest of this talk, first, second, third, fourth, fifth, before they get to be adults. Here we've taken bugs with and without Cavalironia, and I've highlighted just like how many days it takes to get to the fifth instar. And you see a sort of stark contrast that it takes a lot longer. Development just starts to slow way down if you don't get Cavalironia. And this is much more obvious. This is a box in my lab from a couple days ago. These bugs were hatched from eggs at the exact same time. On the left, lucky bugs got Cavalironia. They're now adults. On the right, we have these bugs that are just sort of stuck in the fourth instar. My lab hates this because sometimes you want an experiment to end and they just won't die, but they also won't make it to adults, and you just have to feed them every Saturday over and over again. But it's a really sort of a very consistent phenotype, regardless of whether we feed them on plants or we can feed them on fruits. It matters a little bit, but in general, you really need to have it. So why is this? So Buchner would say, probably some nutrition-based thing because they're in the gut. Somebody like Nancy Moran, sort of the founder of our field, would say the genome sometimes tell you like what they make. And with Cavalironia, we definitely see that they have some capacity to make some vitamins and amino acids. But part of this is actually more like the fungus growing ants in my mind. So if we look at the gut, which I'm now just sort of strung out here, and we zoom in a video, that ball of stuff in the middle, that is bacteria that's being shuffled back to this thing called the M4B, which is a bulb. And they actually eat the bacteria. So some of it is just coming from, I store these bacteria, and then I'm gonna shuffle some back into this digestive chamber to eat later. Okay. So the next step, because I'm an evolutionary ecologist and transmission matters in terms of the evolutionary dynamics of these systems, is to ask how these are transmitted. I'm a mother. These are critical. I would give them to my kids. Like, that's just what I would do. But we see a ton of diversity across insect symbioses of how organisms pick up and acquire their symbionts. And when we go into the ocean, we see the same thing. So repeatedly, there's many different ways to get to the same end. So we did an experiment many years ago now where we took some bugs, we fed them GFP-labeled bacteria, we reared them to adults, and then we just put them in boxes. My lab's very technical. We've got like boxes and plants, lots of boxes and plants. And then we just started to track the offspring to see if they had the GFP bacteria. The only way they can get these bacteria is some way they got out of the adult and then get picked up by these offspring. What you see here is we have each of the offspring life stages on the x-axis. We've got the proportion of those individuals that have the fluorescent bacteria. We do not see it in the eggs. We don't see it in the first instars. We can go out into fields and screen, just use sequencing to look for Caballeronia in eggs and first instars. We never see it. So this is not getting vertically transmitted directly from mother to offspring. But suddenly in the second instar, we are starting to pick up these fluorescent bacteria, which means a bunch of cool things. Some way it got out of the adults. So one way that could happen would be if it's just getting excreted out. It could happen if it is circulating through the plants, which has been shown in some insects, that you spit them back out again and then other individuals pick them up. But they're certainly picking it up. The other interesting thing is a lot of these bugs that are getting to adults don't have this bacteria, but they do have Caballeronia. So you don't need this transmission mechanism. You don't need other individuals with it in the environment to pick it up. You can, even in our lab conditions, with the soil that we purchase, there is Caballeronia around. You're microbiologists, microbes are everywhere, and they can pick it up. So it is environmentally acquired, sometimes getting it from other individuals. And that happens after the second instar. So they have to go through a molt before they can start to pick it up. First instars do not feed a lot. So some of this may just be a behavioral, like, I don't eat that much. Some other hypotheses in this system or systems like it is that the gut has not developed to a point that even if the bacteria got in there, maybe they just can't establish because the gut's not ready for it. So how does this happen? So this is Jason. And I feel like everybody has a science superpower. We all have a science weakness too. Mine's writing. I'm a really slow writer. But his superpower is really natural history observation. So if you went onto a nature walk, you would see him doing this a lot. Right? He looks around a lot. And in the lab, he does it too. And he noticed that nymphs seemed really attracted to this brown, sticky, gross stuff that the adults occasionally excreted. This is not their normal feces. We see it just every once in a while, but they really seem to be feeding on it. And he walks into my office, and Jason always has a million billion ideas. And he's like, I think maybe this is where they're getting their symbionts. So he has a million billion ideas. So myself and his co-advisor are like, let's put that on the list of the many things that you can look at. But the first thing that we did is to look and see if the symbionts are in there. And they are in there in pretty high density. We can culture them out again, but it's pretty hard. That may be because of sort of plastic changes on the part of the bacteria to protect themselves. It may have something to do with this, we think is just this proteinaceous matrix that is protecting them, including protecting them from us. But we can culture them and get them back. And then Scott joined the lab. Scott worked on feather lice of birds and their evolution before coming to my lab as a postdoc. And he's like, oh, I can do a behavioral assay on anything. And so we did the classic thing, which is just choice assay. So we put this sort of fecal excrement on one side that has the symbionts in it, and we put nothing on the other side, and then just like set up a video camera and watch what happened. And here that is. And what you see over here, I don't know what you can see back there, is right there. A bug just sort of walked over there and seems to be feeding on it. That thing in the middle is just a piece of squash. So that's like home plate where they start with. And over the course of this video, which is sped up, they're not quite this crazy, you see many individuals go over to that squash or go over to that fecal spot. And I think during this video, in particular video, you see no bugs go to the other side, which leads to a significantly strong preference for the side with the fecal spot. And we can get the same results if it is just bacteria alone, though it's not as strong, suggesting that there's something about the feces itself that is attractive, but they are attracted to the bacteria alone. And we can also get it, if we're really patient, we can raise adults without symbionts. And we can get them to produce the species, which is very hard because most of them die before they're adults. And they are attracted to the feces without symbionts as well. We will not be repeating that experiment. And they feed. So here you can see that this individual is probing into the surface of the fecal spot, presumably to pick up symbionts. Of course, there's lots of nitrogen in there. There's all kinds of other things. So I can't say that it is just the symbionts that they're getting, but this leads to really high transmission success. So when this is the only available option is this fecal spot, a lot of the individuals do pick it up. And in these trials, they were there with the fecal spot for 24 hours, right? So the fact that they all don't get it may just be that if we had waited longer, maybe they all would. Okay, so this is cool. So I just want to orient you. We got 3 plastic boxes, 3 pieces of zucchini, and a bunch of baby bugs. And the box on the top has a little fecal spot, right, which is right there. And the 2 boxes on the bottom don't. One just is like controlled, doesn't have anything. The other one, we heat killed the symbionts in the fecal spot, or at least as far as we can tell, it killed them. And then this is sped up, but this is about 24 hours after they've been put in this box. And you will notice something sort of drastically different in the boxes on the bottom. They're moving around like crazy. They keep looking. And the box on the top, they're doing what squash bugs do best, sitting there. They just sit there. And what we think is going on is that you put them in the boxes originally. White is with no symbionts, and in black, they had the opportunity to get symbionts from a fecal spot. They're moving around a lot, so they're off the squash a lot. Lights go off, they sit there. Bugs sleep too, by the way. Lights come back on, we see a little bit more movement. And then by 48 hours, we see a lot of movement of these ones that haven't had the opportunity to pick up symbionts. I have no idea why this happens, but it's super cool. Is this some signal from the gut-brain axis? I don't know, but it is something that we really are interested in exploring because it tells us something critical about the evolution of this system, is that it seems like they have evolved these particular behaviors to go look out, find these symbionts, and then get the capacity to know, I'm done. I'm not going to wander around anymore where there's spiders and scary things. Okay, so then once the bacteria are in the host, what happens? My husband is a software and hardware engineer. And when he's doing hardware, he says, sometimes you just have to be the electron. I took 1 year of physics. I have not thought about the electron since. So, but I really want to think about like these individual, a bacterium. What is it like to be like that? This journey that they're about to go on, they just got swallowed or sucked up from a piece of poop. This is the second talk today that used the word poop, by the way. What does that journey look like? So let's follow that journey. So we're going to start at the top of the M4. So the M4 has all those crypts, and then there's this bulb at the top. And right here between the other midgut sections and the M4B is this constricted region. So once the symbionts come in, they come in and it's like the race begins. This is the beginning of the Boston Marathon. They are ready to go. And some of them are starting to move up this channel. So once we see that they've moved up the channel, that constricted region starts to close. But it's only closing in the bugs that have the symbionts, right? Which suggests that there's some cue. I've got these things. I'm good now. There's a lot of animal microbe associations that have evolved to like get the good stuff and keep out the bad stuff. So this might be a signature of sort of balancing those two, of being open for picking up bacteria and then closing the system to prevent competitors of your bacteria or to prevent pathogens from coming in. And so what we see is that when they have the symbionts, these crypts close. These are all sort of second and third instars. This door is not going to stay open forever in the aposymbiotic bugs. So in the ones without Cabrillarinia, by the time they molt to the fourth instar, they have all closed. It's like development's just like slowed down, right? We see that at the whole host level. It's taking a long time to molt, and this constricted region eventually closes. We're not sure if it ever opens up again. There might be periods, especially in between molts. Like, molting's pretty weird. If you wanted to turn yourself inside out again and formed a new version of you, everything's getting sort of messed up. So there may be opportunities that these doors open again, but it certainly seems like they're largely closed. If we move to the bulb, here you see a video, and you can see the individual, some bacteria here. What you'll notice is this is on a loop. They're not crazy, but they are molting. There are some yellow cards up there, but nobody told me what those mean. I have 5 minutes. Okay, so they are moving, so they are mobile. And then once we get to the crypts, remember it looks like this. And they're really mobile. Look at her go. Isn't she cute? I love this. Like, finally, I can be like, look, there's my organism, and I can see what she's doing. I was so excited to see this the day this was taken. They can move about 3 times their body length. I'm a nerd. I did some math last night. So we put this bacteria in competition with the fastest runner of the Boston Marathon. The fastest runner of the Boston Marathon, human, wins, but just barely, right? So, and that's putting them on a course relative to their size. By the way, we all lose. So if we zoom out just a little bit, you can see individuals that are sort of moving through the lumen. This one is moving up into the crypts. And then these ones sort of like their butts wiggling. So what we think is happening is there's microvilli at the opening to the crypts, and sometimes they just get sort of stuck in that. So it might be a mechanism where you sort of along this channel, you eventually get trapped and start heading into these crypts. The crypts start to get bigger, and the bacteria are sort of colonizing along the edges of the host tissue. We don't know if this is biofilm formation, how they're getting attached, but we consistently see this pattern where they're colonized along the surface. The crypts start to get bigger in response to the symbionts. This is not physics does not suggest that this is the symbionts just like putting pressure on them, but they're actively sort of growing in response to the symbiosis. And then the symbionts have to be live for this to happen. So you just not just some bacterial signal that does this. Then the bacteria decide to change, right? So we've seen all these host changes, the crypts are getting bigger and stuff. The bacteria lose the motility. So here we can see a population like really late day 1, and it's just like Brownian motion in those crypts at this point. We do not see any that show sort of active movement. And I'm going to ruin microbiologist, and somebody's going to explain this to me. We start to see some really cool filamentation of the bacteria in some individuals. Some pathogens do this. It's not a phenotype that's known really well, but they start to get bigger. Oops, sorry. And we don't see this in all bugs, but we see it really consistently, and we cannot get it to happen in vitro. And now I have 2 minutes. Okay. So what is the story behind this painting? So this is Peter. He is an Emory undergraduate, and he was partnered with Anthony, who took all those beautiful images, through a program called Science Art Wonder. So this is a program that I started with Emory and Georgia Tech students in 2017. And the idea is that we take scientists and we pair them with an artist, and they have a conversation. The scientist brings lots of images, hopefully, tries to understand the background of the artist and sort of explain the system. The artist makes a sketch, they talk about the sketch, the artist makes a visual arts piece, and then they write a narrative together. The idea is that any science that we project to the public, we want to be accurate. So the scientist plays a part in that. And so what Peter saw in this story, and at that point we only had the host side of the story, is that the host changes in response to this symbiosis. So this is his visual depiction of each of the things in the middle is the crypt, and the color changes because now the symbionts are there. I wish I could have brought this in person, but it's a really amazing painting. All right, I've been told that I don't have any time. Christmas bugs. But I'll tell you one last thing, actually. So in this book, we fed it 2 different bacteria, a red and a green one. And what we notice consistently when this happens is that the population near the posterior end, all those crypts are individually colonized. They only get red or green. Whereas the crypts at the beginning, they look fainter because they have both red and green. And I wanted to show this because Colleen's a little bit of my biology superhero. And she has had some really beautiful commentaries on the idea of mixed infections. This idea of like having all this intra-strain variation, like what does it mean? Does it mean anything? And in this case, what is interesting is within the host, we get places where there's a lot of sort of variation within a crypt and other places where there's single infections. And I think that's driven by this process of just the stochastic process of more and more of these bacteria are getting into the crypts. And so the population's getting less dense towards the end, and you get these individual infections by maybe just one bacterium. So 2 quick take-homes. I think one take-home is that the world's a beautiful place, and that we can really only sort of see the dynamics of this if we try to take the perspective of both the host and the symbionts. And I think that's something that our field needs to do more of. It's really hard, but it actually works really well, which is the second take-home message. When you bring together people who have really diverse interests and backgrounds and abilities to think differently at different scales, you can tell a really interesting story. So with that, I have a lot of people to thank. And if you would like to see more cool science art, you can take a picture of this QR code, and you can see some of the artworks that have been created through Science Art Wonder. Thank you.
Thank you.
Yeah.
Once it has the symbionts. So I was wondering if they are attracted to the fecal spot even after they have that fecal symbiont and their region is restricted.
Yeah, so the question is like a little bit about the timing of this whole thing, right? No, they are not attracted to it anymore, right? So when we see that like once they're in there and they've got it, then they just sort of settle down. Mm-hmm. So they're not moving much. So we don't see any significant attraction to one side or another because they just like, I'm done.
That makes sense.
Yeah. The timing of it is that through that process, so we can see that happen even after just 24 hours, that constricted region is still open. Okay. And so there's a lot of other things that are happening though. We know we can get a priority effect even before that thing is closed. So there's some other physiological changes happening in there, but the constriction region is still open. They could take up more. They just don't because of behavior and some other things that we don't understand.
That makes sense. And then a follow-up question. You mentioned the 4th instar bugs that aren't colonized with the symbiont, so the region also closes. Mm-hmm. Even when that region closes, if you were to put those bugs in an environment with the symbionts, do you think they'd be attracted to them?
Ooh, yeah. So we have not looked at attraction in later instar bugs, and I think that's a really interesting question. Our data suggests that they cannot pick them up anymore, okay. But yeah, I don't know about behavior, and we should do that experiment.
Cool. Thank you so much.
I'm wondering if the bacteria likes to filament when it's anaerobic, maybe the role of oxygen in the gut.
Yes, so the bacteria are anaerobic and do quite well. Like, this is the cool thing. You're like, oh, don't have any incubator space. I'll grow them at room temperature, just out here on LB, and they grow pretty well. So it's pretty easy. We think that there could be a role for oxygen in here. There's a lot of competition. They get pretty dense in those crypts. So in a lot of insects' guts, pH is really important for mediating interactions, so pH may play a role. And then newest thing that postdoc walked into the office, we're like, this is a cool conversation, is that he actually thinks that tryptophan plays a lot of roles in sort of signaling and processes in here.
So
I have a question, sorry.
Yeah.
So the attraction to the fecal pellet is really interesting, and I wondered whether you had thought about using it in an applied way, as whether you could use it as a trap, that you could then attract the nymphs and then try and kill them in terms of agricultural applications.
That would be super cool. I mean, there's a lot of interest from farmers in figuring out a different way to do this. These bugs really quickly gain resistance to pesticides, in part because work by Yoshi Kikuchi, who's a scientist in Japan, has shown that the bacteria can confer antibiotic resistance. And what you've seen here today is that it's really easy for bacteria to move around, and so it just sort of can sweep through. And there's just no control measures, particularly for organic agriculture. So something like that, where you could trap them, would be ideal.
Hi, really cool work. I was wondering if there are Cableronia phage and if you think the crypts are at all involved in quarantining phage infections, if so?
Yeah, I don't know. So Jason, remember the million ideas? Oh, by the way, he's Dr. Chasen now. So even if you have a million ideas, you can get the focus to get a PhD. He did start doing some phage work. So I'm very fortunate that one of my colleagues at Emory is Bruce Levin. He works on phage all the time. He's got an infectious love for both phage and CRISPR. And so we did start looking. We got interested in some phage that are related to a plant pathogen in the system. We didn't find any Cableronia, but also we had to rein Jason in a little bit. So we need to come back to that project.
Well, my real question is, you convinced us you can't beat these microbes in a foot race, but do you think you could do it in a canoe? But you haven't done that calculation, so I won't ask.
I cannot do it in a canoe. I'm very slow.
But I'm actually interested, you showed some data that the insects are attracted to the bacteria even outside the fecal sample, right? So do you know anything about that sort of chemical attraction and how that works?
No, so Jason's actually going to go to the Max Planck this summer to work with Martin Kaltenpoth, and we've started some assays to try to get this. We think it's a volatile signal. It doesn't have to only be volatile, but that's what we're going for first. Yeah, to be determined.
Hi, I was wondering do you know if there is any kind of interaction between the Cableronia and the gut microbiota of the bug that might be as well beneficial? For the bug?
Okay, so I think one thing you saw a little bit of 16S data. There are some other bacteria in there. We have found evidence recently that the Cableronia can facilitate another bacteria getting in there, but it's actually not good for the bugs. So it is a bacteria that seems to be mildly pathogenic for the bugs. So that would be the opposite of what you would want. I have no idea. There's all this Cableronia that's upstream in the other parts of the gut, and we have no idea what they're doing, what their role is, how they're interacting with the other microbes. We just have not explored that at all.
I have another question. I was wondering, you briefly mentioned you changed the diet of the bugs to fruit.
Mm-hmm.
I'm curious what you saw with those diet changes.
Yeah, so squash bugs can feed on both plants and fruits, which makes them particular agricultural pests because if it's the beginning of the season, no fruit, they're fine. If it's the end of the season, the plants are pulled, but there's fruits on the ground, they're fine. And at least under our lab conditions, they do a little bit better when they're on plants than fruits. So we see a stronger benefit of slightly stronger benefit of having the bacteria when you're on fruits. I think because you're starting at a lower baseline. Oh, zucchini. Oh, squash or fruit. Yeah, sorry. Yeah. So when we're raising them on a plant, we're raising them on a whole plant, which is what they're doing in nature most of the time. But once the fruits come out, sometimes that's a better option for them, and so they'll feed on both. Presumably, but I'm not totally convinced that it's sort of the sugars that make them better on the plants. Plants are really nice because once you tap in, they're like, you just have a lot to suck out. And so I think some of it's just easy access to just taking in a ton at once when you're feeding on a live plant.
So the fecal matter is a periotrophic membrane that's excreted by the aphid?
I have no idea what this fecal matter is. As a parent, for those of you who have raised a newborn for that first day, it looks exactly like meconium, this thick, gross, disgusting stuff. But we don't know what its origin is. We don't know its chemical composition. We don't know.
I would imagine that it's a chitin protein mix, right?
Yeah, I mean, I think that's certainly a good hypothesis. Yeah.
All right. Thank you.
Yeah, Connie.
I'm just curious what the nitrogen content of the food that they're getting more and more. Are these bacteria like in aphids supplementing their nitrogen or their amino acid diet?
Yeah, so our group, Allison Ravenscraft, Yoshi's group have all done sort of a little sort of Caballeronia genomics to look at this. And there is some capacity for amino acid supplementation and vitamin supplementation. And I think that is a fruitful place because we can actually potentially in the system knock out genes and then put them back in and see what it is, to see what role that is versus like we just eat some of these cells once in a while. Yeah.
Great. Well, I think to keep things on time, let's thank Nicole. Fantastic talk.